Thank you, Catherine. Thank you all for being here this morning. I'll try to keep to my time. I just want to basically thank also CBB and obviously University of Waterloo in general for hosting this. It's a great time to be here. What I'm going to be talking about is a little bit of Huron's role in some of the biotechnology areas and how it relates to maybe some of what you're looking for and doing, and eventually maybe some innovations as well that may be relevant to everybody. It's really a snapshot of what might be relevant for you, so I'm only going to be touching 3 points. University of Waterloo link with Huron and how Huron can actually advance a little bit of this. Some of our focus, because we're in optical imaging, we'll have to be a little focused, so we're focused on digital pathology, and that is one of our important focus areas. And then a little bit, but not too much, about global research and how imaging and biotechnology imaging can actually help global research. And I'm going to be touching on some of those. It's going to be probably a little disjointed, but I mean, I just want to touch on as many things as possible while still maintaining some focus. So Huron Technology is actually a spin-off from University of Waterloo. 20 years ago, 1994, it started under the name of BPI, Biomedical Photometrics, Inc. We actually have one of our founders as well in the audience here, Dr. Campbell. We have everybody literally at the company is related to the university. So from Savas Chamberlain, our chairman, Ted Dixon, one of the other founders and longtime CEO of the company, Savas Damaskinos, our chief technology officer, and myself, all have been here at the University of Waterloo for a very long time, obviously some longer than others. But I consider this basically our home, and basically everybody here considers University of Waterloo our home. I lived in this building for years. They were about to charge me rent, I was saying. Just so that you don't sort of lose the size of the company. The company is very small, like all Waterloo companies. Starts very efficient and goes very far. Even though we're a very small company, we are punching probably something like 50 times above our weight when it comes to effect. We have 100 clients globally, and you can see from some of the names, I'm not going to point at this one. Some of the other ones are very, very large and reputable organizations that are using extreme science for extreme problems, and they're really using our technology to advance some of that. And we have run-of-the-mill products as well, but that shows you at least how far we've gone, literally from Europe to Australia to everywhere in the US. In fact, Japan as well. Because it's a biotechnology conference and a biotechnology week, I just wanted to sort of anchor that to our biotechnology focus. So I went to Biotech Canada. That's why they defined Biotech Solutions to be basically solutions that would do work to provide better healthcare and preventing disease, among other things that we don't focus on. So we're basically targeting these two top portions of the biotech solutions. I mean, I don't say we're trying to be a biotech company. We were biomedical photometrics. I mean, we are a biotech company, but only on these focus areas. And again, just to situate the company in your mind before we got into a little bit of the biotechnology or technology, the technology that was coming out of the University of Waterloo here from the physics department in '94 has been continuously reaping award after award, international award after international award, way, way, again, more than you would expect from a small company that still remains small today. I didn't go earlier than 2005, but I mean the Frost & Sullivan Technology Award in 2005. That was, again, validated when we went to a scanner contest, international scanner contest in 2012. This is basically the Olympics of scanners. I mean, this is against the GEs and the Leicas and Hamamatsus and the Nikons and all those. And we had basically 2 areas that we competed in, and we cleaned house. We got top prices on both fields that we went in. And then 2013, we had another sort of tap on the shoulder from Frost & Sullivan for best system in digital pathology in the market in North America. And that was literally just by them talking to our customers. It wasn't even something that we had to apply for or go and compete and win. That was them calling us and saying, I mean, we talked to our customers and They're saying you guys are great, and that's why we got this award. It just gives you a little bit, again, of sort of the results of some of the effort that we're making and how far it's taken us. So from the technology, because it is a technology company, I touch a little bit on the science, not much. I promise I'm not going to bore you. It's the whole reason for the company to exist. The raison d'etre of the company is basically this patented piece of optics that started at that time. We labeled as a macroscope, and we have a registered trademark, MACROscope. Your microscope is a microscope. A macroscope is a larger macroscope. And you can see a little bit later, we have 9 patents, and we have 6 more that are coming, that are patent pending. One of them is actually sort of already is crossing the line. One of them in particular I highlighted here, I'm not going to go through details, is potentially going to give the company yet another sort of 20 years worth of tremendous success, huge innovation with tremendous potential for biotechnology solutions, even for cheap, portable, and even sort of remote areas type diagnosis. Back to the initial patented technology, it's basically something that makes us get microscopy level imaging at very macro level. So if you think of a microscope, you're usually looking in the ocular and you're looking at an objective that has maybe half a millimeter by half a millimeter field of view, quarter millimeter. Here we're talking about centimeters of field of view in one time. And some of the incarnations, and we have been 20 years doing this, some of the incarnations of those lenses are on the right there. Again, I promise this is the last, maybe there's one more science slide. It takes an objective and blows it out of proportion. On a normal objective, you have a working distance on a microscope maybe of a couple hundred microns. Half a millimeter, a third of a millimeter, maybe less. Our working distance can be anywhere from 3 to 30 millimeters, to 300 millimeters, from 3 millimeters to a foot, the working distance between the nose of the optics and the specimen. The field of view can be up to 10 centimeters. We can actually image stuff at microscopy level that's 10 centimeters wide. In fact, we've done it once, Savas Damaskinos have done it once with 1.5 meter long specimen. So it takes microscopy and literally blows it 3 or 4 or 5 orders of magnitude compared to what you would know. I'll stop there. So how do we do it? And again, this is sort of a primer, say, on technology, so you can understand later on why we do it the way we do it. If you were to image one small sample, 1 square centimeters, at high resolution, you would probably have a microscope objective, half a millimeter by half a millimeter, quarter square centimeters. And then you end up, quarter millimeter, square millimeter, and then you end up tiling this 20 times on the side. You get 400 tiles and you get your automated microscopy image, which is good, great. Or you can do it the Huron way, where you take the entire strip in 1 square centimeter in one go, one time, and you still have the access to the high resolution imaging that you would have with a normal microscope. So the fundamental difference is that we are not using an objective for the majority of our technology that's innovative. And this is basically the core of it, and you'll see what that can get us towards. Obviously with that, and I just want to touch on some of the problems that we end up facing and some of the problems that we end up solving are not just physics and optics. In order to build systems, we have a ton of data. We need to now to literally create a Google Earth from every microscope slide. If anybody is using any of those, you're going to be using a microscope slide that's sort of 1 inch by 3 inches. We can create an entire Google Earth out of every single one of those. We literally generate a football worth of data, football field worth of data for every small specimen, where you can see every sort of little piece of grass. And that's what we can get. That's the kind of richness of data that we have. So we have a lot of data management behind that. We have a lot of data acquisition. We have a lot of image analysis and compression and management and stuff like that. So even though it's a physics-initiated technology, it's not just a physics problem. It's a comprehensive, multidisciplinary field. Again, just to situate where we're doing the imaging, now with this kind of technology, we cannot go, I heard some people are in atomic force microscope. We don't go to that level. We don't go into sort of telemetry with sort of meters dimensions. In the spectrum of biomaterial, if you wish, a human body is like in the 1 meter sum. And at the other end, you have in the nanometer, we basically span this entire range. It's not a small one. It's actually a very large range because it spans whole organs, in fact, whole human organs, and we'll touch on that a little later, like whole human brain, all the way down to DNA, not DNA strands, but I mean DNA sequences. Very, very quickly, whole specimen microscopy. So you can get from one tissue, you can get a slide that has multiple spinal cords, and instead of going to only the one field at a time only, you can actually get that anywhere across the entire specimen. Same thing with prostate. Prostate cancer, they basically slice it, prostate into 6 pieces and put it on 6 different slices, but then they don't combine it together. Now we can combine the entire image by image, the entire prostate in one. That gives you the range of specimens that we can do, and this is actually from one of our customers. This is your normal microscope slide, and the rest there is a whole organ. We have a variety of products that can target all those, and the 2 that I want to touch on are Widefield and Fluorescence, and I skip a little bit of that. Our focus is on digital pathology, so we apply this technology now into digital pathology field, and these are the areas in digital pathology that we can address, and all with the purpose of providing better patient outcomes. And this is something that is valuable today, and I'm going to show you some horrible examples and what we can do towards them. You might have heard of some or all of those, but I mean, I can guarantee you that somebody that you know is going to have been affected by some of those scandals in pathology, by misdiagnosis and all this. The reason for all those, and this is, as you can see, from 2002, since it has been tracked for 10 years, it has been going on, it has not stopped. Not enough pathologists. A pathologist's work starts with the lab, where you get the slides and you put them into those folders, eventually to go to the pathologist. But they're still doing it in a very manual tracking methods. And you can see the errors. You cannot really tell what's what here because of the manual transcriptions. At the back end of pathology, that's what happens. And the reason I have a busy slide here is to show you that this is not just one guilty person. This is everywhere. This is everywhere. That's what your decision, the decision for somebody's diagnosis is going to be decided on. Plenty of error potential here, and digital pathology will help that. Telepathology is where you have no pathologist in that area, in remote area, like Sudbury or Timmins, and then you have some expertise that's concentrated somewhere else. You can actually use digital pathology to do remote diagnosis. Archival. You have 100-year-old buildings that were really built well, where the floor is caving because the amount of glass that they have to keep is so heavy, and they have to push it. All this can be translated into digital pathology, and it will go away. Education. This is how pathologists learn. 6-headed microscope, 8-headed microscope, but with digital pathology, they should be able to basically get proper education, like here, and testing and stuff. So we are doing this now on a whole organ. So this is a whole breast from Sunnybrook. For diagnosis, I mean, not only for learning, but I mean, even for cancer diagnosis, trying to find the margin on where the tissue, where the cancer has penetrated or has gone. I'll use this point to switch to also cancer diagnosis in fluorescence. I touch on fluorescence only. You can actually detect where the cancer is growing and not by using fluorescence techniques. I'll skip on the fluorescence itself. If you need to understand a little bit more, I can tell you, but this is basically higher energy. Imaging modes, a special imaging mode for multiplexing. It can be used for everything else other than biology, but we are using it for imaging cells, not just all the other stuff. But we can expand into all the other stuff. Our fluorescence imaging is Confocal, which is an added version, added value for imaging fluorescence, and it allows basically for much higher resolution. One of the unique things that our technology allows us to do well is the fact that we can multiplex imaging very, very, very fast. With that, you can build structures of tissue and identify different structures of tissue from a whole organ in a very precise and specific way. Tissue segmentation allows now to build whole organ models based on 30 of those labels and get entire architecture. This is a mouse. I'll skip on the 3D. You can now combine the bright field and the fluorescence, and then you can, again, superimpose processes that are only possible to see with the combination. And you move now from rodents to human, because now we can actually do that. Instead of the 100-year-old human brain map, we are actually building modern human brain maps that are millions of times more precise. And I'll show you a little bit here. I'll just leave you with the 20 seconds video here. Instead of 50 microns and 100 micron resolution, we can get to sub-micron resolution now on a whole 3D brain model that you can use now to map. Last but not least, because we have been in existence for so long, we have customization that is basically in our DNA, if you wish. We have built instruments for all over the world with all sorts of capabilities that are not just digital pathology, but these are all the capabilities that we have, and particularly for that. We say if you dream it, we can build it. So unique and old technology are very, very innovative, award-winning. We are facilitating digital pathology. We're not responsible for making it, but we're facilitating it. We're advancing global research in the human brain mapping. We're very open for collaboration, particularly with our links with the University of Waterloo. And we say, I mean, if do you have any problems we can solve, maybe we can actually do something together that is unique. With that, thank you very much. That's it.
We have time for a couple questions for Réda. Yes?
First of all, I'd like to thank you for a nice presentation. And then my question is about the source you are using. That's right, you are based on your presentation, you are using the fluorescent technique. So are you using the infrared for the illumination? If it's infrared, then are you the sensor, is it the sensor, infrared sensor or digital sensor? Could you explain about the source and the sensor?
So we have, the answers to all of the above is yes, literally. We have illumination from the UV, like in the 400 nanometer or 380 even. And the longest illumination that we have, longest excitation we have is in the 800s, 820 or something like that. And we have emissions that are spanning into that 850. So depending on the applications, we choose the illumination. Laser or not laser, we have not yet used tunable lasers. Nobody has asked us to, or the solutions to any problem was not requiring any of that yet. The detection is rarely extending beyond that. So we don't extend beyond the far sort of infrared, if you wish. NIR is usually something that people would request from us as an add-on. Standard, we use RGB, like I mean up to 600 nanometer, 650 nanometer, but we have gone to the 800 and 850. Answer your questions a little bit?
And so just quickly, if I understand correctly, so your sensor are in the range of infrared and digital?
The detection happens even all the way from 400. Sometimes we excite with 380 and we image in the 400 nanometer. And when we excite, obviously, with the 700 or 650 or 800, we have to have detectors. For the fluorescence, we use photomultiplier tubes that have a very, very wide range, and we select the ones that are targeted in that band, in this band of the spectrum that we image. So it is flexible. We have multiple choices to do. I think that was one question.
Any other questions, Hamid?
So it's always good to hear that the resolution and the quality of the images you are capturing is increasing. But the main question is, and we see that in other fields of biomedical, CT, MR, 700 billion images in the US alone annually, maybe more. We are close to 1 trillion images. And everybody gets worried about the storage, which is also a legitimate concern. But who should process all this?
Absolutely.
Because we don't have intelligent software to do it, and we don't have many doctors, many experts. I'm sharing a general process,
Yeah.
and not your process.
Absolutely. No, absolutely. And you're definitely touching on one of the areas that now is having to take center stage because it was not before. At least I can think very quickly of two ways. One of them is actually one of the collaborations we have with, potential collaborations we have with one of the researchers here at the University of Waterloo, I think with Catherine's group, is basically selective sampling. Even though we can get to a very high resolution, we can do selective sampling and intelligent sampling. So you can actually only get the data that is going to be relevant so that you don't waste your time with the 90-some%, maybe 99% of the data that's useless. The other thing is that we are now ourselves trying to find ways of targeting the computational, real-time computational decision-making to decide what data to store, even if we sample everything, what data to store and to store it in what format on the fly. So real-time processing before what I say, bury the data to disk. Because once you bury the data to disk, you really have to exhume it, and it takes longer to get it than to save it. Because it needs to be structured and da-da-da. So before you bury the data to disk, just milk it for everything you know you're going to need, store that quote-unquote compressed version or the high-value version, and then leave the data then later for longer-term storage and da-da-da. You may want to exhume it later. It will take forever, but you would have at least gotten the best value very quickly. Storage is becoming cheaper, but I mean, the data generation is far outstripping the storage sort of decline in price. So smart sampling and smart processing are the two ways that we are at least thinking of and trying to do.
Okay, thank you very much, Réda.
All right, well, thank you.